Well, hello everyone. Sorry for not speaking French. But today I'm going to tell you a little story in my master's project related to a brain region called the hypothalamus and Alzheimer's disease. So let's first start with Alzheimer's disease. It is the most common form of dementia, and when we hear about this disease, first time we think of, it's probably memory loss. So actually it's one of the main symptoms of the disease, and that's why the study of neuroscience has centered in brain regions related to the control of memory, such as the hippocampus and cortex. However, patients often present also systemic alterations such as body weight disorders, alterations in lipid and glucose metabolism, disorders of sleep, and hormonal dysregulation. All these systemic alterations are known to affect also the development and progression of Alzheimer's disease, and they have one thing in common. They are all controlled by a brain region called the hypothalamus. So this makes now the hypothalamus a very interesting and important region, but yet unexplored in the context of Alzheimer's disease. So let's talk a little bit about the hypothalamus. Here I want you to observe where it is located in the human brain. So the hypothalamus, it's in the base of the brain. Here you can see it. And it is surrounding a structure called the third ventricle that you can see here. This structure is filled with cerebrospinal fluid that in a previous talk we saw that it's a liquid that contains molecules and information from different parts of the brain. But then how can the hypothalamus work? Well, it needs the function of different cells, and we're going to see looking closer at its anatomy. So at the cellular level, well, we know that the brain contains neurons, right? But there are multiple of other cells that help neurons accomplish their function, such as sensor cells, as I call them here. You can see that microglia, it's one of them. This is the main immune cell in the brain, and it's in charge of sensing everything that's happening inside the brain. But also in the hypothalamus, we have a very special cell type called the tanycytes. These cells are located surrounding the third ventricle and therefore are able to sense all the metabolic molecules coming through the cerebrospinal fluid. Lastly, we have endothelial cells that are forming the structure of blood vessels and therefore can sense molecules coming through circulation. Then all these sensor cells gather the information and send signals to neurons that are the ones that can regulate metabolism directly through the release of hormones and neurotransmitters. But as I said, even though the hypothalamic functions are altered in Alzheimer's disease, the role of this brain region has been ignored. For this reason, the objective of my project is to determine if the hypothalamus is altered at a pre-symptomatic stage of Alzheimer's disease. Why before symptoms appear? Because we know that at the end of Alzheimer's disease, all the brain is affected, including the hypothalamus. So if we want to know if this brain region has a role in the disease, we need to see changes at early stages. So for this, first I asked if there were changes in functions related to the hypothalamus. For doing this, I used the mouse model of Alzheimer's disease. So it's a mouse where we can see some of the characteristics that we find in Alzheimer's patients. Since we're interested in a pre-symptomatic stage, we selected an age of 3 months. These mice developed symptoms, cognitive symptoms, after 6 months, so this is considered an early stage. Why using a mouse model? Because we cannot study actually pre-symptomatic stages in humans. So first, I evaluated the body weight of these mice, so a function controlled by the hypothalamus, and actually found that there was an increase in the body weight of the Alzheimer's mouse. This was accompanied by an increase in the accumulation of fat and adipose tissue in the abdomen, and then also extracted from plasma, the extracted plasma from these mice, and evaluated the hormones that are regulated by the hypothalamus. Actually, here we also found alterations in hormones related to metabolism and to stress. So with the first approach, we saw that there were physiological changes in functions related to the hypothalamus. But now we wanted to see if actually something was going wrong inside the hypothalamus. So we analyzed the different cells that are composing it and that are controlling the physiological functions. For this, again, I used the same mouse model. I took specifically the hypothalamus from their brain to analyze the cells that are contained in it through a technique called single-cell RNA sequencing. But what is this technique? Let's imagine that we have any brain region, here the hypothalamus, and the hypothalamus contains millions of cells. So what we do is that we have to cut it small and small and small and small until we obtain individual cells. So we have individual neurons, individual sensor cells, and now we can ask if each individual cell of Alzheimer's disease has a different gene expression than the healthy mouse. So with this technique, we obtain the following map, where each of the dots that you see here represent one of our cells, and it contains the information of all the genes that that cell has inside it. With this, we were able to identify all our sensor cells and also neurons, and then start asking biological questions, such as if there were changes in the gene expression of specific cell types in the hypothalamus at a pre-symptomatic stage. So with this here, I'm showing you a graph with the number of genes that are changing in each one of the cells, and what we actually found is that the sensor cells were the ones showing early alterations in their gene expression. But this still doesn't tell us much, so we wanted to go deeper and analyze the biological processes related to these changes. Why do we want to check this? Because by identifying and understanding the pathways, the processes that are changing inside the brain, inside the cells, we can design therapies that can target this and be able to improve the quality of Alzheimer's patients. So here I'm showing you a list of the different processes that are altered in my sensor cells, but I don't expect you to read all of these, so I group it to make it easier. So I have a list of processes all related to cell stress response, so how cells respond to stress, that are increased in all my sensor cells. I have another group that it's specifically decreased in endothelial cells related to immune response. But then I had an interesting one that was specifically very increased in tanycytes that was related to inflammation via a molecule called interferon. So we got interested in this result because it's the first time that it has been described for Alzheimer's disease in this cell, and tanycytes are very have a big role in controlling both metabolism and hormones. So we wanted to go deeper in this analysis. And our technique allows us to do this. So now we can go and only take the tanycytes that are, as I said, are sensing everything happening in the cerebrospinal fluid and ask about their subpopulations. So biologically, the tanycytes are divided in two groups. Here in the lateral wall are the alpha tanycytes, and here the beta tanycytes. So we were able to identify this computationally and now ask if the changes that we saw in inflammation were specific to one of these subpopulations. So here I'm showing you 30 different genes that are related to this inflammation response, and in red you can see when they are very increased. So we see that all of them are very increased in beta tanycytes, but not in the alpha ones. So if you remember, the beta tanycytes are located in the ventral part, so we identified that specifically these cells there were showing a very strong inflammation. So after being able to understand this, we can conclude that we found alterations in the hypothalamus at a pre-symptomatic stage of Alzheimer's disease at both a physiological and at a cellular level. At the cellular level, if we go back to the initial diagram that I showed you, we saw that the sensor cells specifically show changes at early stages of the disease that we hypothesize that will send signals to the neurons, impairing their function, that will be then shown by changes in the physiology, such as body weight disorders and the previous one that I showed. Specifically, we saw this interferon response inflammation in tanycytes, and now I mentioned before that we wanted to know this because of finding therapies. So now the question is, if by blocking this interferon response, we are able to have benefits for patients with Alzheimer's disease. So for that, we need to go back to humans. So I said we use a mouse model because we cannot study pre-symptomatic stages in humans, but we want to give treatments to patients. So actually, for making sense of these results, a recent study found that interferon was increased in the cerebrospinal fluid of patients with Alzheimer's disease since early stages. And as a perspective of what will be my PhD work, we have now access to hypothalamus samples from postmortem brains from human patients with Alzheimer's disease and healthy individuals. And we will perform the analysis to try to find something related to the changes observed in mice. With that, I want to thank everyone. And yeah, I'm able for questions.